Hey everybody, my name is Mason Hansey, and this is my PowerPoint on flow cytometry, and let's get started. So a quick overview of flow cytometry. This is a technique in which a flow cytometer can detect and measure different characteristics of different populations of cells. Some different measurements that this can deal with are the total DNA, different surface receptors, the transient signal, cell size, among others. This is very fast-paced and a very fast-paced and rapid test, and it can quantify data at high rates and can differentiate different properties in a cell. It allows for purification and analysis while in suspension. Like I said before, it allows you to pick out very minute details of the cell and allows us to find little different characteristics about it. So, information of flow cytometry. The device is made up of three different components. There's the fluidic system. This is the transport of samples from the sample tube into the flow cell and past the laser. There's the optical system. This is made up of separate objects like lens and optical filters, which can alter wavelengths and analyze different wavelengths that determine cell type. There's also the electronics component. You can see that down there with the laser light source. This can analyze the cell cycle and its properties and can cater treatments and diagnosis based on the cellular makeup of the cell. It is extremely specific in this pursuit of obtaining cell information. You'll see we have different types of like flow cytometry, and I'll touch on that right now. With FACS, this is fluorescence activated cell sorting. Like I said, this is known as fluorescence activated cell sorting and is unique in flow cytometry. In this system, it caters the sorting of heterogeneous mixtures of cells at an individual level with the help of light scatter in the cells. This provides a more in-depth view of the cell and its characteristics. It also provides two different data points between qualitative and quantitative data. This is an even more specific and fine-tuned approach to flow cytometry. It can be catered to different situations and in ways in which cell can be sorted. This means whoever is working this FACS sorting system can predetermine the standards on which the cells will be characterized, so he can kind of cater what he's looking for. It almost acts like a filter, if you want to think about it that way. Scatter plots. This is information put into scatter plots. This information is put onto scatter plots that can signify each main event and compare multiple parameters at the same time. There are different intensities that are represented across different axes. Similar cells and cell makeups will be plotted in a closer and tighter group. On the contrary, the scatter plots will be spread out and have less correlation depending on the similarity that the different cells or intensities hold. We see that this is best used in multi-parameter data, and still individualizes each dot based upon its reaction. This isn't technically a actual flow plot, but this is essentially what a scatter plot is. You can see close-knit groups and also really spread out ones. Finally, the immunologic use of flow cytometry. This allows us to understand the molecular anatomic makeup of many different pathogens and antigens, knowing the characteristics such as proteins. Genetic makeup of different proteins allows us to cater different immunizations and medications towards fighting these pathogens. Specific cell types and its characteristics is vital in understanding how to best defend ourselves and in our bodies from possible immunologic pathogens. Like I said, it's like making a map, and you could follow that, and you could also break down exactly what is attacking us, and we could find weaknesses in it, or in many pathogens through this. And these are my references. Thank you.